In this video, we'll look at how tissue culture has revealed how plant root or shoot meristem fate is determined. First experiment I'll point out is that first experiment in which it was demonstrated that parenchyma cells are totipotent. Totipotent means that the cells can become any part of the plant and in fact generate a whole new plant. In animals, only embryonic stem cells have the property of totipotency. There are many animal cells that are pluripotent. For instance, blood cells that are blood stem cells can form either white or red blood cells, and muscle stem cells can form either skeletal or smooth or cardiac muscle, but only muscle cell, except for the fact that I told you recently that they can also become brown fat. Anyway, totipotent means it can become anything. Pluripotent means it still has some choices, but that those choices have been narrowed to a subset of all choices. This is a little different when we talk about what happens in plant tissue culture, because you're talking about a set of cells that already had a job within an organism. These were cells, lots of them were storing nutrients in that big carrot root. Some of them were photosynthetic cells from the carrot leaf or stem. All of them were parenchyma cells, meaning all of them had only primary cell walls, not secondary cell walls. And these results are just part of the evidence that tells us, because many experiments have followed this, that plant cells can de-differentiate. They can take on a fate, a role in the organism, and then, given the right environmental circumstances, they can abandon that role, abandon that fate, and de-differentiate, become willing to listen to a new set of signals and take on a new fate. So, broken up carrots, single cells of carrots, can form carrot embryos, which then develop into whole new carrot plants, which are, of course, of the same genetics of the parent plant, and so they're considered clonally reproduced carrots. Another example of de-differentiation and taking on a new fate is sadly amply well demonstrated when you try to do some weeding. Now, weeding very often just turns into an act of weed propagation. You dig into the soil, you try to get the root, but you cut the plant in half. And the top half of the plant you throw away, and it roots, it forms new roots. Those shoots generate new roots. And the bit of root that you left in the soil, it doesn't just die, it forms a new shoot. But what should be interesting to you about that is that the set of cells that were shoot cells abandon shoot identity and say, root is what is missing, I had better form a root. And the cells of the root that are now on their own make the decision to form a replacement shoot. How is this computation carried out? The evidence that I ask you to look at to figure this out is that auxin is a hormone made primarily in the shoot apical meristem and delivered by a transport system dedicated to auxin transport that moves auxin, A-U-X-I-N, down the plant. So it's coursing from the shoot towards the root at all times. And then there's another hormone called cytokinin. Cytokinin that's always moving up the plant, generated in abundance in the root apical meristem and then delivered through the existing transport system up towards the shoot apical meristem. If I then make a cut in a plant, what are we going to see? Here's a cut in a plant. Cytokinin is going to move from the bottom of the plant into that wound site. Auxin is going to move from the top of the plant into that wound site. And the plant is going to get, the cells are going to get the signal that, hey, there's a cut here, but I'm attached to the root, I'm attached to the shoot, I better just proliferate and fill in that space. And the cells that do that proliferation, the cells that have been released from the context of being part of an intact plant and are now in a new environment, are called callus cells. Callus cells, given the signal of lots of auxin, lots of cytokinin, and essentially the same level of concentration, reproduce quickly and form a big callus, a big collection of that cells, lots of them. If you just provide them with nutrients and not the hormonal signal, not some auxin and some cytokinin in equal proportion, then they just sit there. They stay alive, but they don't reproduce. When you have removed the shoot part of the plant from the root part of the plant, the callus cells that form at the top of the root are getting the cytokinin signal still, but they're not getting the auxin signal. And they successfully interpret that as there is an intact root, there is not an intact shoot. When I redifferentiate, I had better form a new shoot apical meristem, which will then form a new shoot, because that's the part that isn't present. Conversely, if the source of cytokinin is removed, all you've got left is a shoot. Plenty of auxin is going to be delivered to the callus that forms at the wound site. No cytokinin or very little cytokinin is going to be present for those cells. And they'll recognize the fact that more auxin, less cytokinin means there's a lack of a root, and a root apical meristem will form.